Hey everyone, and welcome. We are about to go on a journey through the incredible evolution of vaccines. And you can think of it like a technological arms race, right? We've gone from showing our immune system a really blurry photo of a criminal to giving it a perfect high-definition 3D hologram. So let's dive in.
But okay, before we get into all the different generations of tech, let's just start with the most fundamental question of all. How does a vaccine even work? How does it get your body ready for a fight that hasn't even happened yet?
You know, the easiest way to think about it is this. A vaccine is basically handing your immune system a wanted poster. It's a sneak peek of the bad guy, the virus, the bacteria, so that if the real one ever shows up at your door, your body's defenses go, aha, I know you, and can take it down immediately. And as you're about to see, the way we make that poster has changed a lot. Which brings us right to Generation 1. The approach here was, well, about as direct as you can get. To make the wanted poster, you show the immune system the entire criminal. No messing around. So, the very first vaccines used the whole virus or the whole bacterium. Now, scientists did one of two things. They'd either weaken the pathogen so it couldn't make you really sick, that's what we call live attenuated, or they would just kill it completely with heat or chemicals, and that's called inactivated. Either way, the immune system gets a really good, long look at the entire enemy. And what's so amazing is that this foundational technology is still so important today. I mean, it all started with Edward Jenner's smallpox vaccine way back in 1796, and then it was refined by legends like Louis Pasteur, and today, it's still the core principle behind vaccines we all know, like MMR and polio. But, you know, using the whole criminal for the wanted poster, it has some drawbacks. It's not always the most efficient way. So scientists started asking a new question. What if we don't need the whole person? What if just one unique identifying feature is enough? And that question, that's the key to Generation 2. This is where things get really clever. Instead of the whole pathogen, scientists figured out how to isolate just one specific piece of it, a key protein, which we call a subunit, or maybe a toxin that a bacterium makes. This was a huge leap forward in both precision and safety.
And the tech that made this happen on a huge scale was recombinant DNA, which really hit its stride in the 1980s. This was a game changer. For the very first time, scientists could take the gene for just one viral protein, pop it into something simple like yeast, and turn that yeast into a little factory that just churns out perfect copies of that one specific part.
So, you end up with two main approaches here. For bacteria that create a poison, like tetanus, we can make what's called a Toxoid vaccine that trains your body to fight that specific toxin. And for viruses, we can make protein subunit vaccines that use a key piece of their outer shell. The Hepatitis B vaccine was actually the first one ever made this way, a true landmark in medicine. Okay, deep breath. We've gone from the whole criminal to a key piece of evidence. Generation 3 is where the story takes a radical science fiction kind of leap. The question became, what if, instead of giving the immune system the finished wanted poster, we just give it the blueprint and let our body draw the poster itself? And that is the world-changing idea behind third-gen vaccines. We're no longer injecting the what, the protein or the pathogen. We are injecting the how, the genetic instructions, DNA or RNA, that tell our own cells how to build that protein. Our own body literally becomes the vaccine factory. The biggest advantage here? Speed. I mean, this changes the entire playbook. You don't need to spend months or years carefully growing a dangerous virus in a lab anymore. All you need is its genetic sequence. You can design a vaccine on a computer in a matter of days. It's a complete paradigm shift, from physical manufacturing to programming with pure information. And there are a few really clever ways to get these instructions delivered. You can tuck the genetic code inside a different, harmless virus that acts like a delivery truck. That's a vector vaccine. You could use a little stable loop of DNA called a plasmid. Or the one we all heard about recently, you can use messenger RNA, or mRNA, which is like a temporary self-destructing text message that tells our cells what to build and then disappears. So where could we possibly go from there? If Generation 3 is about giving our bodies the blueprint, Generation 4 is about becoming master architects, designing perfect synthetic immune targets completely from scratch. This is the absolute cutting edge. Instead of just making a copy of a natural protein, scientists are now designing and building highly specific synthetic little pieces of proteins called peptides, or they're using nanoparticle platforms to build perfect mimics of viral structures. Remember that 3D hologram? This is it.
We're creating a flawless piece of evidence for our immune system to study.
And this is not just a theory. This tech is already out there working. The HPV vaccine is a perfect example. It uses something called virus-like particles, basically empty shells that look exactly like the virus on the outside, but they're completely harmless because there's nothing inside. This is where the future is headed. Platforms that could one day give us a universal flu vaccine or even teach our immune systems how to hunt down and destroy cancer cells. Okay, so let's just take a step back for a second and look at this entire journey from start to finish. This timeline really puts the acceleration into perspective, doesn't it? Look at those gaps. It took over a century to get from the first to the second generation, then just a few decades between the next leaps. The move to recombinant DNA in the 80s was massive, but the recent explosion of genetic and nanoparticle platforms, the pace of innovation is just breathtaking. And this table really is the perfect summary of the whole story. First gen, the whole pathogen, the entire criminal. Second gen, purified parts, a key piece of evidence. Third gen, a huge leap to just the genetic instructions, the blueprint. And now, fourth gen, synthetic design, where we build a perfect 3D model of the target, like the amazing VLP technology in the HPV vaccine. It's such a clear and powerful journey when you look at it. We've gone from using the actual physical pathogen to using nothing more than pure information. It's a story of ever-increasing precision, safety, and of course, incredible speed. Which leaves us with one last, really fascinating question to think about. Now that we can essentially program our immune system with a simple set of genetic instructions, what other instructions can we give it? What other diseases could we teach it to recognize and defeat? The possibilities are really just beginning to unfold. Thanks for watching.